Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Last chapter, we kind of walked through the structures of what makes up a cell, and this chapter, we're going to jump right into what those structures do. And this week, we're going to start with the structure and function of that lovely little plasma membrane. As always, right here, you'll see our terms and definitions slides kind of popping up around me. Feel free at these moments to go ahead and pause the video to grab the vocabulary we will be using today. Or you could pop on over to my Patreon, the link is down in the description below, and grab my PDF slideshow for absolutely free. However, if you are a patron, you get access to this video and the accompanying materials a day early. In the coming weeks, we'll also be working on reworking some of the Patreon as well. So keep in tune for that and also in tune for some things on the website. But without further ado, let's talk about plasma membranes. Today, we'll begin our discussion of the plasma membrane by talking about the fluid mosaic model. And what that is it's basically the current model being used by scientists to describe the structure and makeup of the plasma membrane. And basically what it says is that you have this kind of mosaic of protein molecules sort of suspended or embedded in this fluid phospholipid bilayer. Now, if we remember about phospholipids, what they're made of is this hydrophilic or water-loving head and the hydrophobic or water-fearing tails. So you get these kind of back-to-back double layer membrane with those water-fearing hydrophobic tails tucked in the middle of these hydrophilic heads protecting them. The plasma membrane is what is known as selectively permeable, which means it can let certain substances through while keeping other substances out. So an easy way to think of this is when you wear a mask on your face, it allows the oxygen and carbon dioxide through the material. However, the droplets coming out of your mouth and nose are not allowed through that fabric, as well as any viral agents or other pesky little things in those droplets to get through. So again, those respiration gases get through, but everything else gets caught by the fabric. And that's more or less what selectively permeable means. So how exactly do things get through this membrane? Well, we're going to begin with a quick discussion of passive transport. Now, the big deal with passive transport is that it doesn't require the input of energy to happen. So the big example here is what is known as diffusion. Now, diffusion is simply movement down a concentration gradient. So you'll get this area of high concentration, and these molecules or particles or whatever it is will end up moving further and further out where their concentration is lower. So if you look at the images I have here on the slide, the top one shows you just a single particle diffusion across some sort of barrier or membrane until they reach what is known as equilibrium. You have an equal amount of stuff on either side of this membrane. And then the bottom one is essentially the same thing, just with two different colored particles until an equilibrium is met. And diffusion is going to be the most basic form of active or passive transport that we see. So the kinds of molecules that can diffuse freely across the membrane without any help from anything are going to be very small, non-polar molecules like oxygen and carbon dioxide. And that's because it has to be able to get through that very non-polar hydrophobic section in the middle where the tails are. So things like ions and other polar molecules that have charges require transport proteins to get through that barrier. But we'll talk a little bit more about that in a few moments. Continuing our discussion of passive transport, we're going to focus a little bit on water specifically. So when you're having water diffuse across a semi-permeable membrane, there's a very specific word for what that is called, and that is osmosis. So again, osmosis is the diffusion of water across the semi-permeable membrane. So the image I have here is a very common image that's shown in almost every single biology text or chemistry text or any text talking about diffusion and osmosis. So in this hypothetical situation, what we have here is this little U shape with water and some solute dissolved in it. And in the middle here, we have a semi-permeable membrane that only the water can get through. So supposedly the little holes in it are big enough for a water molecule to get through, but not whatever is dissolved there, which is pretty common. A lot of labs can set up experiments like this. It's not hard to get a hold of materials like that if you're in a lab setting. But what you'll notice is there's a higher concentration of whatever is dissolved in the water on one side of that membrane. So after enough time, the water will diffuse across, and the water level on the right side of this U actually ends up being a little bit higher than the left, because an equilibrium is reached and the concentration of that solute then becomes the same on either side. So I know all that is a little bit hard to grasp, but the idea of that is if you have kind of a lot of salt on one side of some sort of container, something blocking it, and barely any other salt on the other side, when you're looking at it is you have a high concentration of water on the side with less salt and a low concentration of water on the side with more salt. So the water is just going to diffuse down its concentration gradient from the high concentration to low concentration. So instead of thinking of these particles moving through the water, think of the water as moving through the particles. That's probably the easiest way to think of it. So just like in the previous slide, you saw particles moving from high to low, the water is just moving from high water concentration to low water concentration, which is going to be important as we move on to our next topic here. Because water does diffuse from these areas of high and low concentration, just like other molecules, just like other particles and substances, solutions, or what cells are in, are going to have what's known as tonicity. So you'll have solutions that have the potential to move water into or out of a cell. And we're going to talk a little bit about that right now. If you have a hypotonic solution, what you have here is a solution with a relatively low solute level compared to the inside of the cell. So there's a lot of water, there's not a lot of stuff dissolved in it. However, the inside of the cell, comparing it to that solution it's suspended in, there is much less water and there are many more little solutes and particles floating around in there. And because that will now set up a water concentration gradient, the water outside of the cell is higher in concentration than the water inside of the cell. The water is going to then move into the cell. And depending on what kind of cell you're looking at, you're going to have different outcomes. Now, an animal cell is going to become lysed or burst, because if it intakes too much water, its plasma membrane is not going to be able to hold it anymore. So basically the cell explodes, the cell is dead. However, a plant cell, they become what is known as turgid. So they become more rigid and they have that lovely cell wall that helps them hold that excess water in. Now, of course, there are limits to everything and it's not impossible for plant cells to burst as well. But what you'll have is a completely different situation where plant cells like to be in slightly hypotonic solutions and animal cells will pretty much burst in a hypotonic solution. But if the kind of water concentration inside and out of the cell is pretty much the same, everything's at equilibrium, that's what's known as isotonic. So that solution that the cell is suspended in is equal solute levels, equal water levels to what is inside the cell. And for an animal cell, they kind of want to hang out in those isotonic solutions where everything's at equilibrium, everything's going to be balanced. For plant cells, isotonic is fine, but they become what is known as flaccid. They're not quite as sturdy, not quite as kind of puffed out, but isotonic is okay, it's fine. But when you get into a solution where there's many more salts and other solutes dissolved in that water around the cell, so the water concentration inside the cell is higher, what you end up getting is the hypertonic solution. So less water outside, but more solutes outside. Now, animal cells and plant cells kind of have the same reaction to this, where they just kind of shrivel because the water rushes out. Again, the water concentration inside the cell is going to be higher than the water concentration outside the cell. And what kind of happens, animal cells shrivel up, they don't have that cell wall there, but the plant cell will still be about the same size as it was in the isotonic solution where it was flaccid, but the inner parts of it will kind of shrivel up within that cell wall. So that's the big difference there. In both cases, it's really not good for the cells. So these cells need to regulate that water balance, and there are ways for cells to kind of protect themselves, but the solution really does have to do a lot with what happens to the cell, because that water does diffuse across those plasma membranes through specific transport proteins for that. But the regulation of that water balance, or the control of the water balance, is known as osmoregulation. So once again, hypotonic solutions have low solute levels relative to cells. Isotonic solutions are kind of at equilibrium, and hypertonic solutions have relatively high solute levels compared to the inside of the cell. I mentioned just a few moments ago that water does diffuse through a transport protein, and this is another form of passive transport. However, it's known as facilitated diffusion. So facilitated diffusion basically just means specific substances like ions and polar molecules need a little help diffusing across that plasma membrane, basically because, remember, the inside of that plasma membrane is nonpolar, so doesn't really like water and charged things coming through there. So you get what's known as a transport protein that will fit a very specific molecule or ion so it can diffuse through without problem. Now, specifically, aquaporins facilitate the diffusion of water, but you don't really need to know the names of other transport proteins unless you're specifically studying that in an upper level biology course. So we've talked a lot about passive transport, but at this moment I'd like to pause a little bit and mention that there's another kind of transport that happens across plasma membranes, and that is known as active transport. The big thing about active transport is that it does require energy in the form of ATP. And why does it require energy? Well, because it's moving things against their concentration gradient. So they're taking things from low concentration, kind of grabbing them up and spitting them out into an area of high concentration. Now, that seems a little counterintuitive. Why would cells want to do that? Well, there are certain situations in which it's beneficial to the cell to have a higher concentration of, say, potassium ions are a fairly commonly actively transported ion. And these ions help in different biological processes that go on inside cells. There are other ions that help with muscle movement and things, and there are what are known as ion pumps. And we will get into all of this in later videos, but a lot of these processes are done through this active transport, and they serve a specific function. So while it does require energy and it is a little counterintuitive to take something from low concentration and move it into a higher concentration, there is a reason. We will get into that later. For all I care about you knowing right now is that active transport is a thing. And active transport, low to high, and requires energy. Whereas passive transport is high to low and does not require the input of energy. The last thing we're going to talk about in our exploration of the plasma membrane today is ectocytosis and endocytosis, which, I get it, those are two very big words and you're probably a little frightened right now. But we're gonna back up. They're actually fairly simple processes. So they're basically how cells transport large molecules and other things across that plasma membrane. These are molecules and other things that are too big to really have a transport protein to help facilitate the movement of them. So if you remember when we were walking through the structures of the cell, we talked about these little things called vesicles, which are kind of like little transport pockets. Ectocytosis takes a vesicle containing some sort of substance. That vesicle will bind to the plasma membrane and then spit out whatever it was carrying outside of the cell. Now, endocytosis is pretty much the exact opposite of that. So it's for the transport of these large substances into the cell. And there's two specific types of endocytosis that we're going to talk about real fast. Now, phagocytosis is the first one, and it's essentially some sort of cellular eating. You'll see this and you'll get kind of these little arms reach out of the plasma membrane and engulf some sort of food in the form of a macromolecule. Sometimes it's another cell, depending on what we're talking about. But it'll engulf that, and then a food vacuole will end up forming on the inside of the cell, and it will carry off into the inner portions of that cytoplasm for digestion and whatever else the cell needs to do with it. Now, if you look at the image here, you'll see some more like terms for like what those little arms are called, the pseudopodium, but that may or may not be important for you to know. Some biology teachers really emphasize that. Some of them just say, just know that it engulfs food, and that's the important thing. But it's kind of a fun word to say, so I just thought I'd highlight it. So the other type of endocytosis here is receptor-mediated endocytosis, which is a mouthful. But basically what it is, it has these specific little receptors that bind to specific substances. Once enough receptors in a small area have been bound to by the specific substance, a vesicle will form and the substances will be carried off for whatever they need to do inside that cell. Well, that's all I really have for you today. This has kind of been a long one, and the chapter 5 videos might be a bit longer than the last chapter just because this chapter is a little bit more technical. We're talking about actual functions. But thank you for sticking with me. Again, if you want my slides and any other study material that goes along with any particular sections, please pop on over to my Patreon. That link is down in the description below. And the day of the video, all that stuff is up absolutely for free. I would like you to be able to study with whatever tools you need. I'm here to help because I understand that for some people, biology is scary, but I promise it's just a lot of big words. So I hope that you have a wonderful rest of your day, afternoon, evening, night, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.